Welcome in. It's incredibly good to have you sitting down here with us today.
Yeah, definitely welcome.
I know you're someone who is intensely curious about the world, someone who genuinely wants to understand the hidden, unseen mechanisms making our planet tick.
Right.
But, and I completely relate to this, that you probably don't want to get bogged down in impenetrable academic jargon.
Oh, absolutely not. Or feel totally overwhelmed by just the sheer volume of information out there.
Exactly. And that is exactly why we are here. You are joining us for a custom-tailored deep dive into a world that is completely invisible to most people.
But absolutely vital.
Vital to the ecosystems of the far north, yeah.
It really is a shadow kingdom. I mean, we were talking about a world operating right beneath our feet.
Literally beneath our feet.
Right. A microscopic and macroscopic network holding the entire forest together.
And our mission for this deep dive is to map that shadow kingdom. We are going to explore Alaska's unique and largely hidden fungal and lichen biodiversity.
It's a massive topic.
It is. Over the next hour or so, we're going to uncover the secrets of highly specialized endemic species that exist nowhere else on the planet.
Nowhere else.
We're going to investigate the mysterious, almost explosive growth of morel mushrooms in the chard, smoking aftermath of massive boreal wildfires.
Which is just a wild phenomenon.
Oh, it's mind-blowing. And we are going to unpack the shocking genetic secrets hiding inside the cells of what is arguably the world's most recognizable mushroom.
To do this properly, we've pulled together a really robust stack of sources for you.
A very heavy stack.
Yeah, physically and mentally. We're looking at complex genetic studies conducted by the U.S. National Park Service.
We've got a fascinating, incredibly detailed 2014 research paper on lichen systematics.
Right, that was published in the Bryologist.
Yes, and alongside those really dense scientific papers, we're also pulling from the Alaska's Mushrooms Field Guide.
Just to get a sense of what this actually looks like on the ground.
Exactly. Plus, we have a deeply detailed USDA Forest Service report on post-wildfire mushroom harvesting.
Which reads almost like a Wild West history book.
It really does. And several comprehensive ecological reports focusing on the Peel River watershed and the ancient prehistoric region of Beringia.
It is a massive stack of material. I mean, if you printed it all out, it would probably break your desk.
But don't worry, we've gone through every page. We've synthesized the most critical, mind-bending takeaways just for you.
And what's fascinating here is the sheer scale of the unknown.
Right.
To put the scope of this deep dive into perspective for you in the state of Alaska, biologists have a pretty great handle on the macroscopic wildlife.
The big stuff.
Yeah. They've definitively cataloged 112 species of mammals.
Okay.
They've identified 525 species of birds.
Right. We know the birds.
We know exactly how many freshwater fish swim in those rivers, down to the subspecies in many cases.
But the fungi?
The number of fungal species could easily be in the thousands.
Wow.
Perhaps tens of thousands. We are quite literally only just beginning to scratch the surface.
And as we'll see today, some of these species have been hiding in a state of deep genetic isolation since the last ice age.
Completely unknown to science until just a few years ago.
Let's start right there, actually. Let's set the stage by rewinding the clock.
Okay, let's do it.
Because to understand what is happening in the Alaskan soil today, you have to understand what was happening there tens of thousands of years ago.
You really do.
I want you to picture the Pleistocene epoch, specifically the period known as the Last Glacial Maximum.
We are talking roughly 20,000 years ago.
Right. Most of North America is absolutely buried under massive, crushing, mile-high ice sheets.
Mile high.
Yeah, think about places like modern-day Chicago or New York City.
Right.
They are under thousands of feet of solid ice.
It's just a frozen, seemingly uninhabitable landscape across the entire continent.
And this defies all logical expectation. There's this massive region stretching from eastern Russia all the way across Alaska and into northwestern Canada.
This area is known as Beringia.
Beringia. And despite being incredibly far north, it is surprisingly and crucially largely ice-free.
That is such a critical point to internalize.
Yeah.
When people hear the phrase Ice Age Alaska, the immediate mental image is a solid block of translucent blue glacier.
Right, just a complete deep freeze.
Exactly.
Yeah.
But Beringia wasn't a barren ice-covered wasteland at all. It was actually a highly complex, dynamic mosaic landscape.
So what did it actually look like? Like, if I'm standing in the middle of Beringia 20,000 years ago, what am I seeing?
Well, you'd be looking at a landscape where the dominant terrain was something called an arid steppe tundra.
Okay, steppe tundra.
Picture vast, dry, incredibly cold plains.
Right.
But they aren't empty. They're dominated by hardy grasses and herbs.
Just as grassy.
Grassy, yeah. And interspersed within that vast, seemingly endless grassy steppe were these crucial mesic or moist pockets.
Okay.
Topography played a huge role here. In the valley bottoms, or along ancient riverbeds where moisture accumulated and was protected from the scouring winds.
Because the wind must have been brutal.
Oh, absolutely brutal. But in those protected pockets, you had resilient patches of conifer and deciduous forests.
So actual trees survived.
Yes. You'd see stands of spruce, pockets of poplar, dense thickets of dwarf birch and alder holding on in the lowlands.
It sounds almost like a cold weather Serengeti.
That's a great way to picture it. And because this massive area wasn't crushed under the ice sheets, Beringia served a dual purpose in the biological history of the planet.
Right.
First, and this is the one most people learn about in grade school, it acted as a colossal land bridge.
The Bering Land Bridge.
Exactly. For millions of years, as global sea levels dropped because so much of the Earth's water was locked up in glaciers, the shallow seafloor between Asia and North America became dry land.
It was the ultimate biological crossroads.
Right, this is where the megafauna come in.
Yeah.
You had immense herds of animals moving across this steppe. You had early horses, massive steppe bison, woolly mammoths, and even tiny creatures like voles migrating eastward from Asia into North America.
And heading the other way.
Passing them on this continental highway, you had lineages of marmots, camels, and shrews moving westward from the Americas into Asia.
Okay, so the land bridge aspect is famous. We've all seen the museum dioramas with the mammoths.
Right.
But the sources for today's deep dive focus heavily on the second role of Beringia,
Which is vital.
which frankly seems way more important for understanding the fungi. The sources keep referring to Beringia not just as a highway, but as a refugium.
Yes.
Can you break down what that means in this context?
Absolutely. A refugium is a glacial refuge. It's a sanctuary.
Okay.
Because Beringia remained largely ice-free while the rest of the North American continent was smothered, it became a safe harbor.
A safe harbor for what, exactly?
For the boreal and Arctic organisms that were already living there, they didn't just passively hunker down and survive the freezing temperatures.
You didn't just sleep through it.
No. They actively adapted, they evolved, and they diversified over millennia.
I want you, the listener, to really imagine that scenario for a second.
It's intense.
Imagine an entire ecosystem, a localized population of plants, animals, and specifically, for our purposes today, fungi.
Right.
Trapped in a localized safe zone.
Yeah.
You are surrounded by impenetrable, towering walls of continental ice to the east and south.
And freezing oceans to the north.
Exactly. You are stuck there for tens of thousands of years. What actually happens to a biological population when it is completely cut off from the rest of the world for that long?
If we connect this to the bigger picture, this kind of isolation acts as a biological and genetic pressure cooker.
A pressure cooker.
Think about how evolution works. When a population is isolated in a refugium like this, it can't mix its genes with outside populations.
Because of the ice.
Because of the ice. There is no fresh genetic material flowing in, and nothing is flowing out.
So they are stuck with each other.
The organisms trapped there are forced to adapt to the highly specific, localized conditions of that sanctuary. The soil, the microclimates, the specific available host plants.
And what's the end result of that?
The result is the creation of what biologists call Beringian relics.
Beringian relics, meaning they are relics of this specific time and place.
Exactly. These are organisms that, today, display significantly higher genetic or morphological diversity right here in this specific region of eastern Beringia, modern-day Alaska, than they do anywhere else on Earth.
Wow.
The genetic data buried in their cells tells the story of their long, deeply isolated survival.
Okay, let's unpack this by looking at a specific, incredibly famous example from the National Park Service study we have in our stack.
The iconic one.
I'm talking about Amanita muscaria, which is commonly known as the fly agaric.
Oh, yeah.
Even if you don't know the Latin name, you know this mushroom.
Everyone does.
If you close your eyes and picture a mushroom from a fairy tale book, or from a certain incredibly popular video game involving plumbers jumping down pipes,
Right.
you are picturing the fly agaric. It has that iconic, vibrant red cap covered in little white wart-like spots.
It is the absolute quintessential toadstool. It's deeply embedded in human folklore.
Right. Now, if you look at a standard field guide, Even the Alaska's Mushrooms Guide we're referencing, it tells you that Amanita muscaria is a widespread species.
Very widespread.
It's described as circumpolar, meaning it's found all around the northern hemisphere, in Europe, in Asia, in North America.
Right.
To the naked eye, if you're just walking through the woods, it seems like just one big, happy, uniform species of mushroom scattered across the globe.
Yeah, a fly agaric in Scotland looks exactly like a fly agaric in Siberia.
Which looks exactly like a fly agaric in Alaska.
But here is the massive genetic plot twist that the National Park Service researchers uncovered.
The plot twist.
When they actually took these mushrooms into the lab and looked at the DNA sequencing data, they realized that this single species of fly agaric is actually masking incredible cryptic diversity.
Cryptic diversity, meaning diversity that is completely hidden from the naked eye.
Exactly. Underneath that identical red and white exterior, there are deep, ancient genetic divisions. Well, the researchers sequenced the DNA of fly agarics found across Alaska, and they discovered that in this state alone, there are at least 3 genetically distinct, completely non-interbreeding clades or lineages of the fly agaric.
3 of them?
And they're living sympatrically.
I want to make sure I have this right. Sympatrically means they're living right next to each other, sharing the exact same geographic area, right?
Right.
So they are in the same forest, maybe even drawing nutrients from the exact same soil, but they are not mating with each other.
Precisely. From a biological standpoint, they are entirely different species walking around in identical disguises.
That is wild.
They have diverged so much genetically that they can no longer reproduce with one another, even when they're neighbors.
And it gets crazier when you look at where these specific lineages come from. The sources detail the ancestry of these 3 Alaskan clades, and it reveals something astonishing about how they got there.
It really does.
Yeah.
The genetic markers show that 2 of these Alaskan clades, which researchers designated as Clade 2 and Clade 3, are what we classify as Eurasian lineages.
Eurasian.
Yes, their genetic closest relatives are found in Europe and Asia.
Okay, what about the third one?
The other lineage, Clade the first, is a North American lineage,
Right.
closely related to the fly agarics found down in the lower 48 states.
Right.
Now, here's the mind-blowing fact that the study highlights. Those 2 Eurasian clades of fly agaric do not exist anywhere else in North America outside of Alaska.
Wait, hold on. You're telling me 2 of these fly agaric lineages in Alaska are fundamentally Eurasian, but if I go down to Washington State or over to Maine, they just don't exist.
They do not exist there.
How is that physically possible if the Bering Land Bridge disappeared underwater over 10,000 years ago? How did they get there, and why didn't they spread to the rest of the continent?
That is exactly the right question to ask, and it's the central mystery these researchers were trying to solve.
Right.
To figure it out, the scientists used a technique called coalescent analysis.
I saw that term in the study, coalescent analysis, and honestly, the math in that section made my eyes cross a bit.
It's heavy math.
So what does this all mean in plain terms? How does looking at a mushroom's DNA today tell us where it was 20,000 years ago?
Think of it like this. Coalescent modeling is essentially a way of reading the DNA of current populations as a historical clock.
A clock.
Every time an organism reproduces, there's a tiny chance for a genetic mutation. Over long periods of time, these mutations accumulate at a relatively predictable rate.
Okay, that makes sense.
By looking at the DNA of these different clades today and comparing how many unique mutations they have, scientists can mathematically trace those genetic differences backward in time.
They run the clock in reverse.
Exactly. They run it in reverse to see exactly when these different populations coalesced or shared a single common ancestor.
Oh, I see. It's like taking an ancestry DNA test, but instead of finding out your great-grandfather was from Ireland, you're finding out your mushroom lineage split from its European cousins 50,000 years ago.
That's a perfect analogy.
Yeah.
And when the researchers ran this coalescent analysis on the Alaskan fly agarics, the results were definitive.
What did it show?
It proved that these endemic Alaskan sequence types, these specific Eurasian clades, were genetically isolated from their ancestral populations in Asia for a very, very extended period.
Meaning they crossed over the land bridge a long, long time ago, long before the last glacial maximum.
Yes. And here is the crucial takeaway. During the last glacial maximum, when those mile-high ice sheets pushed down across North America, the native fungal populations in places like Canada had to slowly migrate south.
Retreating ahead of the crushing ice shields to survive.
Right. But these specific Alaskan fungi didn't migrate south. The coalescent data shows they didn't mix with the southern populations.
Or what did they do?
They survived locally. They rode out the entire ice age in place, hunkered down inside the ice-free Beringian Refugium.
And this isn't just a fun piece of trivia about a pretty red mushroom.
No, not at all.
The sources make a point to emphasize that this solves a massive, long-standing geographical mystery about the Ice Age itself. Because Amanita muscaria isn't just any fungus.
Right.
It is an ectomycorrhizal fungus.
Yes, that is the linchpin of this whole discovery.
Let's define that for a second. An ectomycorrhizal fungus is one that absolutely requires a living host tree to survive.
It can't just live in the dirt.
Right. It doesn't just decompose dead wood. It actually wraps its mycelium, its underground root-like threads, around the living roots of a tree.
They form a symbiotic partnership.
The fungus acts like an extended root system, foraging for water and hard-to-reach soil nutrients, which it pipes directly into the tree.
And in exchange,
The tree feeds the fungus vital sugars and carbohydrates that the tree creates through photosynthesis. The fungus literally cannot live without a living, breathing host tree.
exactly. The fungus and the tree are inextricably linked.
Oh.
So the fact that this highly specific tree-dependent fungus survived the last glacial maximum locally in Alaska provides major, undeniable scientific evidence for something paleobotanists have debated for decades.
It proves that the trees were there.
It proves that fragments of the boreal forest, actual living pockets of spruce and birch and poplar trees, must have survived the deep freeze in Beringia.
Because if the trees had all died out, the fly agaric would have starved to death.
Precisely. You don't get ectomycorrhizal fungi surviving for tens of thousands of years in a grassy steppe. You need the forest.
Wow.
So the DNA of this little red and white mushroom acts as a living fossil record, proving the survival of ancient forest refuges that have otherwise completely rotted away and disappeared from the geological record.
That idea of a fungus acting as a biological time capsule is incredible.
It is.
But that's a big, flashy mushroom that everyone recognizes. Let's transition to something much harder to see.
Okay, let's unpack this.
Let's look at the overlooked microscopic world detailed in the 2014 paper from the bryologist. The researchers here were looking at Crustose lichens.
Yes, shifting from mushrooms to lichens is a great way to show how pervasive this Beringian isolation was across all types of life.
For those who might not know, Crustose lichens are the ones that look like they are literally painted onto rocks or tree bark.
Right, they form a trust.
They don't have leaves or stems, they just form a thin crust. And the paper details a major discovery in Katmai National Park, which is down in southwestern Alaska on the Alaska Peninsula.
A beautiful area.
Researchers were surveying the area and discovered a brand new species to science, which they named Rhinodina pallidescens.
Rhinodina pallidescens.
And it is completely endemic to southern Alaska.
Finding a new species is always exciting, but to picture Rhinodina pallidescens, you really have to recalibrate your sense of scale.
You have to look incredibly closely.
Extremely closely. The paper describes it as having a very thin, pallid gray, sometimes slightly brownish thallus.
The thallus being the main vegetative body of the lichen.
Right. It is cortically, which is a botanical term meaning it specifically grows on the bark of living trees.
Okay.
The researchers found it clinging to the bark of elnus or alder trees, populus or poplar trees, and salix or willow trees.
So it's basically a grayish smudge on a piece of tree bark.
Basically.
It sounds incredibly subtle. How do you even know you found a new species when it looks like a discoloration on the wood?
That is where the methodology of lichenology becomes so fascinating. You can't just identify these in the field.
You have to take them back to the lab.
The researchers have to carefully shave off a microscopic piece of that bark, take it back to a laboratory, slice it unimaginably thin, and look at it under high-powered microscopes.
And when they did that with this gray smudge, it revealed some truly eccentric, defining quirks.
Very eccentric.
Okay, I have the quote from the paper right here, and this is where I need you to translate for me.
Sure.
The researchers noted it was distinct because it has, quote, physiotype ascospores with a prominent torus. I'm not going to lie, that sounds like a spell from Harry Potter, or maybe a rare car part. What am I actually looking at here?
It does sound like magic, but it's pure biology. Let's break it down. A lichen is actually a composite organism.
It's a fungus and an algae living together.
Right. The ascospores are the reproductive spores produced by the fungal partner of the lichen. They are essentially the seeds it uses to reproduce and spread.
Okay, so they are looking at the seeds under the microscope.
Right. Physiotype refers to a very specific structural shape of the spore named after another genus of lichen called Physia.
Okay.
It means the spore has incredibly thick walls with a distinctive barbell-shaped internal cavity where the actual cell contents live.
A barbell shape.
And the torus. That is a thickened, heavily pigmented, ring-like structure that encircles the very center of the spore, right at its equator.
So under a microscope, these tiny spores look like heavily armored little dumbbells wearing a dark belt.
That is exactly what they look like.
Okay, that paints a much clearer picture. But what really seemed to get the bryologists excited wasn't just the shape of the spores.
No, it was how they actually grow inside the lichen.
Yes, this is the really chaotic part. In most closely related lichens within this family, the spores develop synchronously.
They grow inside a microscopic sac called an ascus.
Right. Usually, all 8 spores inside that sac mature at the exact same time, like a perfectly synchronized biological clock.
But in Rhinodina pallidescens, the development is entirely asynchronous.
Meaning they are all growing at their own pace.
Exactly. It's disorganized. Inside a single sac, you might have one spore that is fully mature and dark brown, while right next to it is a spore that is pale and barely formed.
Just totally chaotic.
Because of this chaotic, staggered development, the sac often aborts some of the lagging spores, resulting in only 4 mature spores being released instead of the typical 8.
That sounds inefficient.
It's a highly unusual, chemically expensive characteristic, and it's one of the primary unique features that allowed them to confidently declare it a completely new species.
And the discoveries in Katmai National Park didn't stop with just that one new species.
No, the researchers were doing a comprehensive survey.
And they also found 2 other rare lichens, Rhinodina buckii and Rhinodina oregana.
Yes.
And the paper emphasizes that finding these specific lichens in Katmai was a massive deal for understanding Alaska's deep ancient history.
It really is, and it's all about something biologists call range extensions and phytogeographic history.
Range extensions.
Let's take Rhinodina oregana as the prime example. Before this Katmai survey, this lichen was mostly known from much further south, down in the Pacific Northwest.
Right.
Now, this specific species is known for having absolutely massive spores compared to its relatives.
Massive on a microscopic scale, of course.
Right, relatively speaking. But in the microscopic world, physics matters immensely. Having massive, heavy spores makes you a terrible, terrible disperser.
You can't just catch a light breeze and float across a continent.
No, you drop out of the air very quickly.
You're not traveling light, you're trying to fly a cargo plane instead of a glider.
Exactly. So finding this incredibly poor dispersing lichen completely isolated all the way up in Katmai National Park, hundreds and hundreds of miles away from its main population, suggests something profound.
What did it imply?
It implies that it didn't just recently blow in on the wind. The researchers hypothesize that this lichen has likely been in the Alaskan region since the Tertiary period.
Wait, the Tertiary period? That predates the Ice Age. Right. We're talking millions of years ago.
Yes. Because it can't travel easily, it must have survived the last glacial maximum right there in place.
Amazing.
It suggests that there were unglaciated coastal refugia places right on the edge of the ocean, like the modern-day Alexander Archipelago or Kodiak Island, that remained ice-free.
So this lichen, alongside the newly discovered Rhinodina pallidescens, just hunkered down on the bark of whatever trees survived in those tiny coastal safe zones.
That's the theory, and it's backed by the physical evidence.
This isn't just a mossy smudge on a tree. It's a living museum.
It really is.
It is a biological time capsule proving that tiny pockets of the Alaskan coast have remained virtually unchanged since before the ice ages.
Just holding on through the deep freeze.
It's just wild to think about these tiny, almost invisible organisms holding a genetic record of millions of years of geological survival.
It really reframes how you look at a forest, doesn't it?
It absolutely does. And there's another highly specific endemic Alaskan fungus I want to bring up from the sources.
Okay, which one?
Because it perfectly illustrates how extreme this adaptation can get. It was described in 1975, and it's called Lesinum alaskanum.
Ah, Lesinum alaskanum.
Yeah.
This is a perfect example of a highly specialized Arctic survivor.
So what is it?
The Lesinum genus is a type of bolete mushroom.
Boletes.
These are the mushrooms that have pores underneath their cap instead of gills. And just like the fly agaric we discussed earlier, Lesinum mushrooms are ectomycorrhizal. They need a host plant.
Right.
But Lesinum alaskanum is a purist. It is an extreme specialist.
It doesn't just partner with any tree.
No, it partners almost exclusively with a plant called Betula nana, the dwarf birch.
Now, dwarf birch thrives in the incredibly harsh, unforgiving Arctic tundra.
Very harsh.
We are talking about environments well north of the Brooks Range where the growing season is insanely short. The winds are brutal.
And the soil is permanently frozen just a few inches below the surface.
And the soil in the Arctic tundra is notoriously nutrient poor. It's cold, it's acidic, and the organic matter barely decomposes.
It is incredibly tough to make a living down there.
So how does this specific partnership actually work? How do they survive?
This is where the biochemistry gets truly fascinating,
Okay.
and where the fungus basically becomes the hero of the tundra. The sources point to intense speculation and ongoing research showing that species in the Lesinum genus possess unique proteolytic capabilities.
Okay, proteolytic. Break that down for me.
Proteo refers to proteins, and olytic means to break down or destroy.
So it breaks down proteins.
Yes, this fungus acts almost like an external stomach for the dwarf birch tree.
An external stomach, that's a great image.
In the freezing, nutrient-starved tundra soil, there's actually a lot of nitrogen which plants desperately need to grow.
But there's a catch.
But it's locked up inside complex, stubborn organic proteins from dead plant matter that hasn't fully decomposed because of the cold.
The dwarf birch's roots cannot access this nitrogen.
No, it's locked away in a chemical vault.
So the tree's surrounded by food, but it doesn't have the key to open the pantry.
Exactly. Enter Lesinum elasticum.
The external stomach.
The mycelium of the fungus secretes highly specialized enzymes directly into the freezing soil. These enzymes are capable of breaking down those complex proteins.
Effectively digesting them outside of its own body.
Yes, and extracting the vital nitrogen. The fungus then pipes this freshly unlocked nitrogen directly into the root system of the dwarf birch tree.
And what does the tree give back?
In return, the tree, which can photosynthesize in the long summer sunlight, feeds the fungus the carbon sugars it needs to survive.
That is a crucial survival trick. It's a highly evolved, deeply specialized chemical hack that allows both the fungus and the tree to conquer an environment that should, by all rights, be entirely too nutrient-starved to support them.
Right.
The tree farms the fungus for nitrogen, and the fungus farms the tree for sugar.
It's an ancient alliance forged in the ice.
That idea of a fungus acting as a tree's survival engine isn't just happening in the ancient frozen tundra, though.
No, it's not.
It actually brings us to something happening right now, in the modern day, in the aftermath of some of the most violent events on Earth.
Here's where it gets really interesting.
Let's shift our focus from the deep ancient past and the slow microscopic adaptations to the dramatic violent present.
Okay.
In Alaska's boreal interior, natural wildfires are a fact of life.
And we aren't talking about small brush fires.
No. We are talking about massive, crown-replacing fires where millions of acres of dense forest can burn to the ground in a single summer.
The scale of destruction is awe-inspiring. It looks like total absolute devastation. Blackened trunks, ash feet deep, nothing left alive above ground.
But out of those ashes comes a phenomenon that borders on the miraculous.
It really does.
And it is the focus of the incredibly detailed USDA Forest Service report we have in our stack. I'm talking about the post-fire fruiting of the Morchella mushroom.
Commonly known as the morel.
Right, the morel.
Morels are the holy grail for foragers. They are incredibly highly prized, choice, edible mushrooms. They have a deep, earthy, nutty flavor, and they look like little wrinkled honeycombs on a stalk.
They're delicious.
Chefs around the world pay top dollar for them. But their taxonomy, how we classify them scientifically, is an absolute nightmare for mycologists.
Why is that?
The USDA report literally refers to them as perplexingly polymorphic.
I love that phrase. Perplexingly polymorphic. It basically means they're shapeshifters, right?
Exactly. They change their physical appearance, their color, the shape of their ridges, their size based entirely on microclimates, soil moisture, and local environmental conditions.
So they look different depending on where they grow.
A morel growing under a burnt spruce tree might look completely different from a morel growing 10 feet away under a burnt poplar tree, even if they are genetically identical.
Right. The sources note that because of this shape-shifting, researchers believe there could be up to 22 endemic Morchella species in North America alone.
Yes.
And they estimate there are 5 to 6 putative or assumed species that fruit specifically in Alaska's interior after a fire.
Putative, meaning we think they're a distinct species based on behavior and some genetics, but it's hard to pin down.
But the harvesters who are actually out in the field doing the grueling work of picking them, they don't use the Latin names.
No, they don't care about the genetics.
They just informally categorize them by color based on when they emerge from the ash. They call them the blondes, the grays, and the blacks.
Which is a perfectly practical system when you're racing against the clock.
Sure.
But for scientists, the really compelling question isn't just what color they are, but how and why they fruit in such massive, explosive numbers after such a catastrophic event.
And it all comes back to their hidden life cycle beneath the soil.
Yes. Let's pull the curtain back on that.
What is the morel doing before the fire? Like before the forest burns down?
Beneath the forest floor, long before a fire ever sparks, the morel fungus exists as a massive, creeping underground network of thread-like mycelia.
Okay.
As this network grows over years and decades, it forms specialized structures called pseudosclerotia.
Pseudosclerotia. Let's unpack that.
Think of them as compact, hardened, incredibly dense clumps of fungal tissue. They serve as underground storage bunkers.
Storage bunkers.
The mycelium gathers nutrients from the soil and potentially from the roots of living trees, as there is strong evidence that morels might be mycorrhizal for part of their lives, and stores those calories inside these hardened bunkers.
So they are essentially banking calories for a rainy, or in this case, a fiercely fiery day?
Precisely. They are preparing for disaster. Now imagine that massive, millions of acres wildfire sweeps through the boreal forest.
The heat is so intense it bakes the top layer of the soil.
The host trees are incinerated or killed by the heat. Suddenly, the morel fungus loses its primary food source and its canopy cover. The entire ecosystem it relied on is dead.
The fungus is suddenly facing imminent starvation.
So it panics.
Biologically speaking, yes?
The pseudosclerotia, those backup batteries kick in.
What triggers it?
The sudden loss of the host tree, combined with the drastic chemical changes in the soil from the fresh layer of alkaline ash, acts as a biological trigger.
It signals to the underground network that the end is near.
Exactly. The fungus uses absolutely all of its stored energy from those bunkers to push thousands, sometimes millions, of fruiting bodies, the morel mushrooms, up through the ash.
It's a frantic race against time. It wants to release millions of spores into the wind to blow away and colonize new, unburned areas before the underground network completely starves to death.
It's literally a do-or-die reproductive explosion.
It is the ultimate evolutionary gamble.
And this biological desperation triggers an entirely different kind of explosion. A human one.
The harvest. I want to spend some time on this because the USDA report reads like an account of a gold rush.
It really does.
Let's look at a specific historical example from the report. The summer of 1991, following the massive 1990 Tiktok River fire.
The Tiktok River fire was a monster. It burned roughly 90,000 acres of forest.
Right. And the following summer, when the snow melted and the ground warmed up, the morels popped.
And the humans followed.
Over 300,000 pounds of morels were harvested from that single burn scar in just a few weeks.
The logistics of that are staggering. It was a massive influx of people.
Buyers and pickers flooded into the tiny town of Tiktok from the lower 48 states, from Canada, from all over the world.
They set up makeshift buying stations right on the edge of the burn scar, weighing baskets of mushrooms and handing out wads of cash.
It was pure Wild West economics. But I was reading through the descriptions of the harvesting itself, and let me tell you, this is not a leisurely romantic stroll in the woods with a wicker basket.
Oh, no.
Harvesting post-fire morels in Alaska sounds like absolute grueling, filthy torture.
It is not for the faint of heart.
The harvesters are hiking miles into destroyed, dirty, ash-covered forests. There is no shade because the canopy is gone.
And every time you take a step, a cloud of black, acrid soot kicks up into your lungs.
You are constantly covered head to toe in charcoal.
And because it's early summer in Alaska, the legendary mosquitoes are out in full, terrifying force.
The harvesters are wearing head nets, sweating in the sun, breathing ash, and staring at the blackened ground for hours on end, looking for dark mushrooms hiding in dark ash.
And you can't just take your time, because the window to harvest is incredibly tight.
Right.
The fruiting window in the interior of Alaska can be as short as 4 weeks, usually peaking right around late June or early July.
It's a flash in the pan.
The mushrooms grow remarkably fast, but if the weather suddenly turns hot and dry, the morels literally dry up on the stalk. They turn brittle and disintegrate into dust before you can pick them.
Plus, you have to be incredibly careful about what you are actually picking, right?
Yes, very careful.
The report mentions several ways nature tries to trick the harvesters. For instance, there is an indicator species called the cup fungus, Geopixis carbonaria.
Ah, yes, the cup fungus.
What is that?
It's a small, orange, cup-shaped mushroom. It fruits prolifically on newly burned sites, often just a week or two before the morels are supposed to arrive.
Okay, so it's an indicator.
Right. Pickers see the cup fungus and get incredibly excited, thinking they've found the perfect spot. But the report notes that it frequently fools people.
How so?
It indicates the conditions are right, but often the morels simply never arrive in that specific patch.
It's a false flag.
Exactly.
But a much more dangerous trick is the false morel. I was reading this part of the report and my jaw dropped.
It's serious business.
What are these false morels, and why are harvesters so terrified of accidentally putting one in their bucket?
False morels are mushrooms in the Gyromitra genus. To an untrained eye, they can look somewhat similar to a true morel. They have a brain-like folded cap.
But they are bad news.
They are incredibly dangerous. They contain a chemical compound called gyrometrin.
And the report notes that gyrometrin is a precursor to monomethylhydrazine,
Right.
which, for the non-chemists out there, is literally a component of rocket fuel.
Exactly. It is highly toxic. It attacks the nervous system and the liver. Just inhaling the vapors while cooking false morels can cause severe neurological symptoms.
So if a buyer spots even a piece of a gyrometra mixed into a harvester's basket, they will often reject the entire haul.
You have to know exactly what you're doing.
It's high risk, high reward, and completely unpredictable.
The yield is entirely ephemeral and chaotic.
The USDA report outlines how a patch of ground that yields a massive bounty of 8 pounds of morels per acre 1 year might yield absolutely zero the very next year, even if another fire comes through.
It is 100% dependent on an exact, unreplicable concoction of soil moisture, the severity and heat of the burn, the timing of the spring snowmelt, and the frequency of the summer rainfall.
Which really highlights how volatile and responsive this entire hidden ecosystem is.
It's a dramatic boom and bust cycle dictated entirely by extreme natural forces. The fungus is just waiting for decades for the perfect disaster to strike.
So we've looked at the deep freezing past of Beringia.
Right.
We've zoomed way in on the microscopic adaptations of lichens.
Yes.
And we've walked through the chaotic, fiery present of the morel harvest. Let's zoom out now and look at the uncharted future of Alaska's mycoflora.
Let's do it.
We mentioned at the very top of the deep dive that while we know exactly how many mammals and birds are up there, fungi are basically the dark matter of the ecosystem.
Dark matter is the perfect term for it.
We know they're there, we know their gravity holds the entire biological web together, but we can hardly map them.
And the National Park Service genetic studies we've been referencing really hammer this reality home.
So.
To illustrate the scale of the unknown, the researchers decided to focus just on one single genus, the Lactarius genus.
These are the milk caps, right? Another group of Ectomycorrhizal fungi that partner with trees.
Yes, they are very common. By simply sampling soil cores and collecting random fruiting bodies across various Alaskan parks, they didn't just find a few new things.
What did they find?
They found 28 putative species groups of Lactarius.
28.
28. And many of those groups formed completely unique, unidentified genetic clades.
Meaning they don't match anything else.
Right. When the researchers ran the DNA through global databases, they got no matches. They didn't align with any known Lactarius species from Europe, Asia, or the lower 48 states.
So they are brand new.
They're likely brand new, entirely undiscovered species, just sitting there in the Alaskan dirt waiting for a name.
So how do we possibly fix this massive knowledge gap? We can't realistically have a handful of National Park Service geneticists crawling over every square inch of a state the size of Alaska, pulling up every mushroom they see.
It would take centuries.
No, the scale of the landscape makes traditional surveying impossible.
This is where a massive shift in methodology is happening. Crowdsourcing science.
Citizen science. I love this part of the sources.
It's the only way forward. The sources highlight incredible initiatives like the North American Mycoflora Project and specifically a regional node called Mycota of Alaska.
And who is doing the work for this?
It relies entirely on the public, regular people, hikers, amateur foragers, indigenous communities, nature photographers. They're the ones doing the legwork.
That's amazing.
They go out, photograph mysterious mushrooms in their natural habitat, meticulously document the surrounding trees and soil, carefully dry the fungal specimens using food dehydrators.
And then what?
And then literally mail these dried mushrooms in Ziploc bags to central laboratories for DNA sequencing.
It is a massive, decentralized, grassroots effort to catalog the dark matter before the ecosystem fundamentally changes.
Yes.
Because, and this brings us to the final sobering part of our deep dive, the ecosystem is changing fast.
Yes, this raises an important, deeply urgent question about the immediate future. we have to consider the impact of climate change.
And we aren't just talking about melting glaciers or rising sea levels.
No, the sources include a pivotal study by Morgado and colleagues regarding how summer temperature increases are directly affecting the moist tussock and dry tundra in Arctic Alaska.
Right, because I think the common misconception is that climate change in the Arctic just means less ice for polar bears.
But the Morgado study shows it's fundamentally rewiring the invisible biology of the soil.
Exactly. The researchers in this study didn't just observe, they manipulated the environment.
How did they do that?
They established long-term experimental warming plots in the tundra. Basically, they built small, open-top greenhouses over patches of wild tundra to artificially raise the ambient summer temperature by just a couple of degrees.
Simulating what is projected to happen over the next few decades.
Right, and what happens to the fungi when you turn up the thermostat?
Well, first, the warming directly changes the above-ground vegetation.
The plants.
With warmer, longer summers, shrubs start to grow taller and more aggressively, shading out smaller plants. Different grasses might move in. The whole plant community shifts.
And because of the symbiotic relationship.
Because the fungi, especially the ectomycorrhizal fungi, are so intimately chemically tied to those specific host plants, altering the above-ground vegetation directly and immediately alters the below-ground fungal communities.
They tracked this using something called OTU richness, right?
Yes.
I want to make sure we're clear on what that means.
OTU stands for Operational Taxonomic Unit. When scientists sequence the DNA of a scoop of soil, they don't always know the exact species name of every piece of DNA they find.
Because so many are undiscovered.
Right. So they group similar DNA sequences into OTUs. Think of an OTU like a barcode.
A barcode.
We might not know the exact Latin name of the mushroom that left the DNA, but we can scan the soil, see 100 distinct barcodes, and know there are at least 100 different types of fungi living there.
So OTU richness is basically a direct measure of genetic diversity.
That makes perfect sense.
So in these experimentally warmed greenhouse plots, what happened to the barcodes?
The researchers observed a stark decline. Specifically, they noted that the OTU richness associated with Licenum, remember the Licenum elasginum, the fungus that acts as an external stomach for the dwarf birch.
Yes, the protein hacker.
That specific genetic diversity actually decreased significantly in the warmed plots compared to the unwarmed control plots.
Wait, so by merely warming the ambient air a few degrees, we are inadvertently shrinking the genetic diversity of the very fungi that keep the foundational Arctic plants alive.
Yes, the dwarf birch might survive the warming, but it might lose its fungal partners, which changes how the entire nutrient cycle of the tundra operates.
We are fundamentally altering the structural architecture of the soil ecosystem before we've even managed to catalog what's actually down there.
That is the core tragedy of the situation, isn't it?
We are running out of time to understand an ecosystem that took tens of thousands of years of extreme isolation to build.
It's a race against the thermometer.
Okay, let's take a deep breath and recap the incredible journey we've been on today.
It's been a long journey.
We started in the unimaginable deep freeze of the Pleistocene epoch, discovering how the ice-free refuges of the ancient Beringian land bridge acted as a biological arc.
Protecting and isolating species while the rest of the continent was crushed under ice.
We peeled back the iconic disguises of the globally recognized fly agaric, revealing 3 secretive, genetically distinct ancient lineages hiding in plain sight in the Alaskan soil.
We zoomed way, way in to marvel at the microscopic oddities of newly discovered ancient lichens clinging to tree bark in Katmai National Park, proving their survival since the Tertiary period.
And we waded through the blinding ash and the swarms of mosquitoes to understand the explosive, desperate, post-wildfire reproductive gamble of the Morel mushroom.
If there's one overarching takeaway from this entire stack of research, it is that the ground beneath your feet is not just dirt.
No.
It is a living, breathing, deeply complex historical archive of survival and adaptation.
And that profound realization leaves us with a lingering, somewhat haunting question to consider as we wrap up.
We now know, proven beyond a shadow of a doubt by the DNA, that the immense crushing pressure and total isolation of the Ice Age is what actually built this biological vault.
The extreme, unyielding cold forced these organisms to adapt over millennia, turning Alaska into a genetic stronghold of unique, hyper-specialized fungal biodiversity.
But today, the Arctic is warming faster than any other region on the planet. The rules of the game are changing overnight.
If extreme, long-term cold built this biological vault, what will extreme, rapid warming do to it?
Will these highly specialized ancient underground networks, these external stomachs and deep soil bunkers, find a way to pivot and adapt to the heat? Or are we essentially burning down a microscopic library before we've even had the chance to read the books?
That is a staggering amount to chew on. Thank you so much for joining us on this deep dive into the hidden, shadowy fungal networks of the North. Keep looking down, keep asking questions, and never ever underestimate the dark matter beneath your feet.